Sheep disease update. Perry Wolff. Something everyone will be interested in.
I think it must be the deal with the secretaries who take the minutes and stuff. The people take the minutes.
For the record.
Working for the county, one is Erie County employee, so it's paying a small portion of their employee salaries. It's just a normal county employee working for state, 3 hours once every 7 times a year. No, I guess that's 20 hours a year. You know?
See. Sheep done 2 days. States that have this one.
Okay.
You're on.
Chairman Lent, Commissioners, Secretary Mayor, thank you for letting me come and give you this update. I think everybody's probably heard by now that we're having a disease event in our Rocky Mountain Bighorn sheep herd in Elko County, Nevada, and I just wanted to give you an update of what's happened so far, where we're at as of today, and what some of our proposals are for the future for looking into this. So, as you can see, we have a little disease event in the East Humboldt and the Ruby Mountains. And for those of you that don't know where we're looking at, it's Unit 101. The East Humboldt's just south of I-80 and Wells. And then the other is over in Lamoille County, Unit 102, south of I-80 and just east of Elko. So that will orient you to where we're talking about. As far as current population estimates, at the time when we started looking into this in the East Humboldt's, the 2009 population estimate is 180 animals, and approximately 140 animals had been visually seen in there since January 1st, 2010. I don't have any updated as of today, but those are beginning of the year. For the Ruby Mountains, the estimated 2009 population was 160 individuals, and those were primarily in Lamoille Canyon because that was where we had the easiest access. There were some other attempts to get into some of the other canyons, but the biologists were either snowed out or fogged out and were able to get a good view of what was of the numbers there. A little bit of a timeline in the Ruby Mountains. We first got a report from our biologist there that an observer had been out there and seen some coughing sheep, and that was in the beginning of December of '09. He went out the next day and actually got a visual on some of the animals. 50 sheep seen, 30 plus were coughing and had the presence of nasal discharge. He found one dead ram that was in a fairly advanced stage of autolysis, but he was able to get some lung tissue from that animal and bring it back in for testing. At that time, he noted that there was a younger ram who was fairly sick, very lethargic, didn't get up during the entire observation period, and he and I spoke, and I suggested that that animal should be harvested so that we could get a fresh necropsy sample to figure out what we were dealing with here. So on 12/13, they went in and harvested that ram, and he was taken to the Department of Ag facility in Elko and received a complete necropsy. There was a thought that maybe this incident was quieting down a little bit, but then they had some more reports that there were further sick animals. So I went out there. A group of us went out there in the beginning of January and tried to get an assessment of the animals. Between the 4th and the 9th, they had found 3 additional dead animals, and that 75% of the sheep, and this was in Lamoille Canyon, were still coughing. There was also one mountain goat that was found dead on the side of the road. He was in a fairly advanced stage of decomposition, but again, they were able to get some samples off of him, and we know that there have been incidences of the mountain goats and sheep commingling up on the Ruby Mountains. So we went back up on the 13th and went ahead and immobilized 2 rams and 1 ewe that we could get close enough to. We did live sampling on those animals, ear tagged them, and administered an antibiotic by the name of Draxxin to those animals. And during that period of time and over the next couple of weeks, 16 additional animals up in Lamoille Canyon were darted with Draxxin. They were not immobilized, they were just free darted. Timeline in the East Humboldts, 12/29, there were reports of 6 sheep in the Clover Creek area. There was 1 dead but heavily scavenged ram found, and there was really only the head, and so we're unsure what he died of. On 12/30, the biologist was able to get into the Trout Creek, Beverly Hills, and Welcome areas and noted that sheep were observed coughing with nasal discharge. We went in on 1/2/10 and were able to locate 5 ewes that were immobilized and live sampled, and also had radio collars, satellite radio collars placed on them, and they were also treated with Draxxin. 1 ram was in severe distress, and we elected to harvest him due to the severity of his clinical signs so that we could get, again, a fresh necropsy and determine what sort of organisms we were dealing with. Between the 6th and the 14th, 4 additional sheep were found dead in that area. Teams of biologists went out, and 30 animals were darted with the antibiotic Draxxin. We thought we were kind of quieting down, and then over this past weekend, an additional 22 animals were found dead. Some probably had been dead between 4 and 7 days. Most of them were very heavily scavenged, so we're not sure how much actual data we're going to be able to get out of those animals. So just as a summary, 33 sheep have been found dead in the East Humboldt Range. 4 animals have received complete necropsies, and we've been able to get good samples from them. 35 sheep have been administered Draxxin, and actually 5 ewes have been immobilized, 4 of which have been outfitted with radio collars. In the rubies, and this is as of just this afternoon, 10 sheep have been found dead, 3 sheep have received partial to complete necropsy, 19 sheep have been administered Draxxin, and 3 sheep were immobilized, sampled, and ear tagged. What we're seeing is primary clinical sign is coughing, nasal discharge is present. You can see on the nose down there on the right, the blood was from the fact that we were administering nasal oxygen during the immobilization, so they do not have bloody noses, but you can see in the other nostril, that's kind of that thick white mucoid discharge coming out. The animals, a lot of them are fairly approachable. They're lethargic and are laying down while other animals are up eating. We've also seen some weight loss in some of the animals that have been sick for a number of weeks, and also, again, we've been finding dead animals. Necropsy findings, and what you're looking at right now is we've cracked open the chest. This was a ram harvested out of the East Humboldts, and you can see his rib cage has been lifted up, and there is pleural adhesions, or kind of a fibrin that is gluing his chest cavity to the side of his lung. And you can see his right lung there in front of the ruler, and it's dark purple, and it should be a nice pink color. So on the histopathology and on the gross findings, we're finding anywhere from acute to chronic, and this would be considered chronic. Fibrinopurulent bronchopneumonia, meaning it's affecting the fibrin and pus and affecting the airway passages in the lungs, and it's consistent on pathology with bacterial pneumonia. This is a young lamb that died fairly acutely, taken from the East Humboldt, and you can see the dark-looking stuff is his right anterior lung lobe. Underneath that is his heart, and then his trachea is going up to the right of the slide. The rest of his lungs are fairly unaffected, but that one right cranial lung lobe there that looks dark is pretty non-functional. What we found from our culture and serology results, we've taken serology and cultures on all the live animals and then just cultures on the dead animals. I don't think it's anything too shocking. With pharyngeal swabs and lung cultures, we've isolated some of the common bacteria that we would expect to find in suspected Pasteurella pneumonia, Pasteurella trahelosi, Pasteurella multocida species, and Mannheimia hemolytica. We've also had some other mixed bacterial cultures that they don't report out what the specific bacteria are because they're in such low numbers. We've taken pharyngeal, nasal, sinus, trachea, and lung swabs for PCR, which looks actually at the DNA of the organism, and those have come back positive on both dead and live animals for Mycoplasma pneumoniae, which we know is a player in the bighorn sheep pneumonia syndrome. We've done some serological titers to a whole host of viruses and other bacteria that we know that these animals can contract. And the only ones that have come up with any elevated titers are to BVD1, which is bovine virus diarrhea, and PI3, which is one of the respiratory viruses. This is not, we haven't seen sky-high titers. We've seen fairly high titers, but we've also seen that in other herds. As far as gastrointestinal parasites and lung parasites, we've seen those parasites, but they're in low numbers, and we don't feel that they're causing any significant effect on these animals. We've also done trace mineral analysis. We've done liver analysis on all of the necropsied specimens where there was still a liver to be had. And primarily selenium is certainly one of the trace minerals that's gotten a lot of attention. It can, in low levels, it can be immunosuppressive, and we know that certainly in the West, many areas of the West are selenium deficient, and anyone that runs cattle or sheep knows that many of the salt mineral, trace mineral blocks and feeds that are provided are supplemented with selenium. Selenium in these groups has been low normal to deficient for liver analysis, and then whole blood analysis we've seen again low normal to deficient in blood tested from live animals. So I think it can be asked, you know, why we decided to treat with this antibiotic Draxin. Also in the news is that there's concurrent bighorn sheep die-off events, disease events going on simultaneously in Montana and Washington. We've been in contact with both of those agencies and their biologists and veterinarians, and found out what they were doing. At the time that we first started having our outbreak, Montana was going through and harvesting any animal that they deemed was sick in an attempt to pretty much eliminate any animal showing clinical signs off the landscape. To date, they've harvested about 75 plus animals. This was during their first outbreak. They're now working on their third outbreak that's occurring this winter in Montana. Talking with Washington, they were basically taking the documenting and monitoring route. They were doing overflights on their bighorn sheep and documenting percentage of animals that were coughing, and then following the spread of the disease. At the Wild Sheep Working Group meeting this past week, they now are going to go in and start harvesting a group of animals that on the other side of the Yakima River and see if they can eliminate it over on that side. So now they're looking at harvesting, and I was contacted by Montana, and I think they might be tiring of shooting animals and are exploring the possibility of treating as well. The other reason that we chose Draxin and even considered it is it's at the time was the only marketed antibiotic for treatment of respiratory disease in cattle and swine, and it's indicated for the ones that we usually deal with, Pasteurella species, the Mannheimia, and also has been shown in cattle to be effective against Mycoplasma species. It is a small dosage, so it was relatively easy to administer to animals, including if we were just free darting them. It's meant to be given as a one-time treatment. There has been cure rates or improvement in cattle and swine from a one-time treatment, and that's the way it's really marketed. It's been used safely in domestic sheep and goats, and it's been used fairly widely across the West In wild sheep, certainly we use it a fair amount in our transplantations as sort of a preventative. And then also, this group is not about to be imminently hunted, so we felt safe that we could actually use something because of any possibility of antibiotic residues. The immobilized animals at the time also received vitamin E, which is just helpful for possible capture mortality and also acts synergistically with selenium. I chose not to give them any injectable selenium like Muci or Boci that you might be familiar with, because many of these animals have access to selenium-treated salt blocks, and certainly you can induce selenium toxicity if you give them an injection and they have high levels. So I felt it was safer not to do that. We also gave them Banamine, and again, anyone that's dealt with cattle or horses is familiar with Banamine. It's just an anti-inflammatory, helps make them feel better, and is a good all-around drug. Reduces fever, inflammation, that type of thing. So that's pretty much where we are sitting as of today. What are some of our proposed next steps? We've been talking to a lot of our colleagues. We've been on the phone with Colorado, Washington, Montana, Idaho. We've presented our sort of laid out what we've done so far and what we think we should do next. And this is our proposal. A plan for actually detailed investigation and further surveillance in the East Humboldt and Ruby Mountains for our Rocky Mountain bighorn sheep starting from today through September of 2010. And I think one of the unique opportunities we have here is this, for any of you that have been up and dealt with this group of sheep, is they're actually fairly accessible. They're not high up on a mountain crag somewhere, so we can actually visualize them and safely get to them and handle them, both safely for us and also for the sheep. So our objectives for our next steps are to compare and contrast some of the subherds up in the East Humboldt and Ruby Mountains and look at those animals. We'd like to evaluate these animals for the presence of respiratory pathogens, general health status, and also their nutritional status, looking at trace mineral levels. We'd also like to evaluate forage quality and trace mineral levels and precipitation history at these corresponding sites. There's a lot of material out there that talks about selenium levels and selenium type and availability with varying precipitation rates, and certainly we've had a fairly wet year this past year and are wondering if that could have impacted the selenium levels, which might have led to a decrease in immune status and possibly allowing some of these bacteria to take hold. We'd like to measure the effectiveness of the antibiotic Draxxin. We certainly don't think that it's practical that every time there's a sheep outbreak that we're all going to run around with little syringes and needles and inject all of our bighorn sheep with Draxxin. But some of this has been tried historically with other antibiotics that were certainly not as specific for mycoplasma and also as easily administered. And I think it's important that not only to see if this potentially might work, but also to rule it out if it's something that might be a detrimental thing to do with bighorn sheep. We want to measure survival, look at body conditions. Some of these bacteria have a chronic carrier state, and they feel many of the biologists and veterinarians feel this is something that we should not allow sheep to get to that condition because, one, they're poor animals on the landscape, and two, they might be sort of in a typhoid Mary type of status. We want to look at residual lung pathology. You know, it's hard to run up the slopes when you only have a quarter of your lung working. See if we're clearing pathogens and changing the pathogen picture of these animals. And also, probably the most important is looking at lamb recruitment for these ewes and future growth and return to healthy levels of these herds. We'd also like to look at the forage quality across other parts of northern Nevada where we know we've had heavy precipitation, but we've also conducted captures in the past couple of months, and so we have really excellent data on the health of the sheep themselves. So it'd be and we have pathogen profiles of these animals so that we could go back and compare what their nutritional status was as far as trace mineral levels and forage quality and trace mineral levels in the forage coupled with precipitation, and compare that to what's been happening in Elko County. How we propose to do this? We'd like to mark and radio collar additional animals. Our colleagues from around the West have shipped in about 20 additional collars that we can put on, and this will give us more animals that we can follow up with, and it will also allow us to establish a control group of animals that are marked but not treated with Draxxin and specifically follow their survival. So our proposal is to go into some of these separate subherds and get our hands on some of these additional animals. We're primarily looking at Rattlesnake Canyon. We have not gotten a good look. We know there's animals in there. They interact freely with the Lamoille group, so we think they're probably affected, but we don't know. We know that they have not, we have not gone in there and actively darted animals with Draxxin, so we'd like to get in there and capture 10 animals. Half of them would be treated with Draxxin, the other half would not. All of them would receive radio collars. Since we haven't found any mortalities from that, known mortalities from that area, we'd also like to get in and sample some of those animals so that we can get a full profile of what's going on with them and a better understanding of if they have the same disease process going on. In Lamoille Canyon, we have 3 that are tagged but not radio collared. If possible, we'd like to go back in there and put collars on another 10 animals. We've got fairly good necropsy data, but it's not complete. We tend toward the rams and the lambs is what we're coming up with, and it would be helpful to have an adult ewe and see what kind of status that animal is in. Over in the East Humboldts, in the Willow Trout Creek, Beverly Hills area, we have 5 animals marked. 4 of them are radio marked. We'd like to capture another 6 if possible and mark them, and then again review our necropsy data, and if necessary harvest another animal again to get fresh quality samples and find out what's going on with these ewes, since this has been heavily skewed more toward the rams and lambs. We want to conduct standard necropsy and sampling on all of our any harvested or sampled bighorn sheep, as well as any fresh carcasses that we find. Like to collect the forage and soil samples and evaluate it for trace mineral levels, protein, digestible protein, and be able to analyze that not only in our affected areas but in some of our non-affected areas. Be able to continue with aerial surveys so that we can monitor what's happening with these herds. We know there's animals, but sometimes we have difficulty finding them. Are they there, or have we had other undocumented mortalities? And then also recapture and resample some of our radio marked animals before they leave the wintering grounds to see, do they still have the same pathogens? Have they put on a lot of body weight? Are they still carrying a lamb? That type of thing before they disperse. We'd like to follow the ewes and lambs through at least the 6 weeks. There's been a lot of reports where lambs start out okay, and then as they reach a certain point later in their lamb life, and they start getting into these nursery groups, you can have these little die-offs that go on and lose all of your lamb crop. So it's important not only to see what hits the ground, but also to follow them out. And then also, when we have that information, pretty much look at what we're going to do from here on out. I think what the take-home message I got from all of my colleagues is that what we really need to do is, this is great, everybody's all excited during this initial outbreak, but what we don't do is do a good job of following up and monitoring. Some of these herds that have had pneumonia outbreaks in the past, not necessarily in Nevada, but in other areas, have had poor lamb recruitment for years and years and years. So do we have carrier animals in there? What's the issue? And what we really need to do is to monitor this long term. And again, I think the particular group of animals really lends themselves to being able to follow up with this information and really get a handle on what's happening with some of these sheep. I put together a budget for this that we pulled together. Again, breaking it down, we kind of condensed it down so it wouldn't go on for page after page. Looking at continuing costs for antibiotics. Certainly the majority of the cost is going to be in for sampling of the animals. Each just for the mycoplasma PCR, each one is a $35 to $50 hit. So when you start looking at a bunch of animals and doing multiple samples, it can add up. Necropsy and histopathology costs and supplies. All of our samples are sent up to the Washington Diagnostic Lab. And even though we can do necropsies in-house, we still have to send the tissues and samples out. Looking at forage and soil analysis, not only for the Ruby Mountains and East Humboldt's, but also for some of the other areas where we've captured sheep over the past couple of months. And then replacement costs, new batteries, replacement collars to those, our colleagues who have lent us their collars in a hurry. And then also a big chunk of cash is for helicopter survey and capture costs to get our hands on animals that do not lend themselves to walking up and being darted. So that's an overview of where we are right now and where we would like to go in the future. Any questions? Yes.
I got a couple.
Yes.
What is the, I don't know much about Draxxin because that's for animals, but what's the bioavailability of that and how long does it last? How long does it take to work and how long does it last?
It's very bioavailable in the studies that they've certainly done, and it was mainly marketed for cattle and swine. So the majority of the published research-based studies we have to extrapolate from cattle and swine. In experimental studies that they did with young calves that were given inoculated with some of these bacteria, they were looking at, and compared to controls, they were looking at improvement within 24 to 48 hours, and in improvements in weight gain and attitude and fever reduction. Most of the studies were carried out for about 14 days before these experimental animals were then euthanized and they did the final workups on them. It is primarily meant to be given as a one-time treatment, although if you do not see adequate improvement, it is okay to go back in 12 days and repeat the injection.
They don't worry about mutating organisms from that?
No, they don't.
Where do they get access to selenium blocks? Are there selenium blocks out in the...
There have been selenium blocks put out there because that is one of the concerns is the low selenium, and I think that Tony Wasley had worked with UC Davis and some of the nutrition folks there in looking at trying to develop a mineral block that would be sufficient, and we'd like to reevaluate that and go back and make sure that our formula is correct and can match the forage quality out there, and make sure that when we're putting blocks out, we're putting the correct supplement out.
Any copper in their liver? Did they find any copper?
No, the copper levels have been within normal range, and certainly the liver levels don't lie. When you look at blood samples, the blood maintains its levels of all these trace minerals until the body's totally depleted. So when we just look at blood, it's kind of deceiving because you could have very little levels in your liver, but the liver levels don't lie. So no, and they've been pretty much right where they're supposed to be. So no evidence of any copper toxicity.
I just want to, for the commissioner, I sat through 2 days at the Wolff Sheep Study Group. Perry and Caleb and Mike was there, and with these other states, and every state's doing something different, but I want to say Nevada is doing more for their sheep than any other state. Montana's culling them out, and the stuff that we're doing is so much better, in my opinion. We're doing more for the sheep than anything, and Perry and Nevada should be congratulated for what we're doing. We're taking a scientific approach, and it's very expensive, and we got a $65,000 bill, and we got to, if anyone has any ideas on that, now's the time to come forth or work on it. But I want to thank Perry and the rest of the staff for what we're doing with the sheep, because we have to make a decision tomorrow on these, on the seasons. And it's a tough decision, and so any information we have is gonna be very valuable to us. But, seeing what the other states do, California's got some die-off in the whites down there in the south end, and, Washington doesn't know what to do yet. They're gonna start doing stuff. They're waiting to see all these groups get there and talk it over. But Nevada is doing the foremost for these sheep than what I saw of any other state doing. And I think we should be congratulating them for doing that. And it's expensive. Look at the helicopter time, and that's why, and it will be brought up tomorrow during the legislative, this is a perfect place where we should be drawing down that heritage money. Whether we gotta buy them a helicopter, we got an emergency here. And we should be able to go into that $5.7 million and get it and get the job done. And that's a problem, but that's what I have to say. So, Commissioner Capurro.
Yes, Perry, it's really kind of disheartening to see some of those figures, because if you have 75% of the herd, if I remember, 75% of the herd that was observed in the East Humboldts had the clinical symptoms, and that the survival rate of that, even with the Draxum, just depends on how far along they were, I guess. And
Exactly.
Secondly, if they're that involved, will the lungs recover using Draxum?
That's a good question. I think the Draxin use in most of the domestic animal species, they're trying to get those animals over shipping fever so that they can get to the feedlot, grow, get fat, and get slaughtered. Whereas we're looking at,
keep 'em alive.
we're looking at long-term productivity. So that is exactly why I'm saying that we need to make sure that we monitor these animals. The more animals that we have collared that we can retarget and compare non-treated animals to treated animals, and we're looking at ways now to potentially use something like ultrasound to be able to evaluate the lungs of these animals, working with hunters to be able to come back and say, okay, I harvested this ram 2 years later, well, heck, I'm seeing he still has lung pathology. I think the best thing we can do is follow this out and see, are we doing any good? Are we not doing any good? Or are we making things worse?
I think it's really important with what you're doing for the next outbreak, where you can get to
Exactly.
it quickly, and you know that certain treatments work, isolation and some other things, besides just going and shooting them.
Right.
But, I hate to put you on the spot. Do you have any feeling about how many more mortalities that you're going to experience out there with those two herds?
Tune in daily.
No, I'm sure there will be more mortalities. We still have a lot of winter to go. These animals are under a tremendous amount of stress from being sick. They don't feel good. They don't get up and eat. So yes, there definitely will be more mortalities. And I hope that through our efforts, what we can do is potentially help put this picture together so that not only we can react sooner the next time an outbreak occurs, which we know there will be, but even look at ways that we could manipulate forage, manipulate something to say, oh, look, it didn't rain, it did rain, blah, blah, blah. Maybe this is we know we're going to get hit, so let's have people on the ground starting December 1st and be right on top of that before something would happen.
Thank you.
Commissioner Rain?
Thank you, Mr. Chairman. I had three things. One is, we brought this thing as selenium blocks. Now, selenium blocks, how widespread is the Endow's distribution of selenium blocks in that area? Is it widespread, a few here and there? Has it been a plan, gone on for years?
I'm going to let Mike address that since I'm...
Or is it just, a sample,
Mike Cox, Nevada Department of Wildlife. The region biologists have been working to put a few out kind of as a test just to see use. patterns over the last couple of years. So it's kind of in an experimental mode.
It was interesting. I'd been talking with some sheep producers actually several months ago, and said, if you we find 6 sheep, they need more selenium. Give them selenium blocks all the time. It's one of Endow's great failings. Have not been putting out sufficient selenium blocks. Now I'm sitting here and I'm thinking, wait a minute, maybe they know something. You got low selenium liver. This sheep producer been telling us for months. We actually had a guy at one of our meetings say that. This sheep producer. Need to be sufficient giving them selenium. That just seems like an interesting coincidence, perhaps, or maybe not. The question on the histopathology and necropsy samples, said you're sending them to another state. What isn't Nevada capable of doing that?
No, we don't
No.
have a histopathology
There's no
service in Nevada.
None of that.
And this is the best one in the, WSU's the best one in the country.
Yeah,
There's
the Washington
Local
Animal
ones.
Diagnostic Lab is heavily invested in working on bighorn sheep issues, and they also have been receiving all the samples from all of the different states, so they have a really good
Some things you could do in Nevada, but
database.
not those. Okay.
Not those.
The lamb ratios said they were very low. What's being observed right now? Do we have more firm numbers on
Correct. It's less than 10
that? In both
lambs per
areas?
100 ewes. In the East Humboldts, for sure. I can't give you an exact number in the Lamoille Canyon, and we're not. There's other animals besides those in Lamoille that represent the rubies, but it does appear low. Also in the general rubies.
Okay, now there are other sheep populations. Have they been? Somebody gone up and checked on the other sheep populations we got?
We've got 80 bighorn herds. We're a little busy. We do have standard times that we classify land recruitment through the air, pretty much on every other year basis.
So
for most of the herds.
You see concern. I mean, if we got these 2 herds, has anybody been out looking at the other herds?
Some we have, some we haven't.
So we may or may not. Okay.
We do have a study that we did. It's published on manipulation of mineral blocks up in Humboldt and Washoe County. I can give you a copy of that paper. I published that back in '08. And we looked at those populations that we treated with minerals and those that we didn't. There was no herd performance differences between the herds. Obviously, the Great Basin and its soils have always, at least as long as man's been around, been low in selenium. We don't have die-offs every year. So it's a multifactorial, very, very complex, but it certainly does contribute to the immune system. And with other contributing factors, it can help create a disease storm, if you will.
I think there's, from what I get out there, there's just no clear answer here. One of the from one of the other states I was talking to, and they're looking at some work where the acid rain is deleting the selenium uptake in plants, and so the plants don't have selenium in them due to the forage, and so it's a real complicated process here. And really, no one has answers. I guarantee you that. And so we're just trying to save what we have, and but trying to say this is a problem it can't be done now. And this is just not our people, but it's all the western states, all the biologists put together. Commissioner Cavan?
Is there any evidence that there's segments of the population that are possibly immune, that have built immunity or genetically immune to these critters?
Well, certainly, there's been a lot of work with that in many domestic species, and when we're collecting, part of our protocol is collection of hair and whole blood that can then be used to look at relatedness and possibly genetic patterns, and actually that's one of the things we were just talking about, but there has not been a full-scale study done to really assess. You get a really good picture of who died, but you don't really get a good picture of who didn't die, because you usually don't mess with those. So, but yes, that's something that that we're going to try to get as much information as we can, with the limitations of not being able to get our hands and get a sample from all of the animals in the population.
I had one other question, Harry. Is there any warnings on human consumption of Draxon, nursing mothers and that? I mean, they put a warning on it for human consumption?
Well, under the Food and Drug Administration regulations, I mean, we are using this as an off-label, species off-label. So if this was happening, you know, anywhere near the hunting seasons, we certainly would not even consider giving these animals anything, because most of these would have a withdrawal time for off-label use of about 90 days minimum. We feel that we can't prevent poachers from going in, but other than that, we feel that we're in a very safe window for not potentially exposing any people to this.
Poachers eat them at their own risk, huh?
Exactly.
Okay, Commissioner Howell?
I heard you mention this dioxin has been used on cattle and swine. We don't have anything that we've used on domestic sheep before?
There is, all of these drug companies are driven by the bottom green buck.
Really?
And I know you find that hard to believe, but you know, the bovine respiratory disease complex is where you make your money. And it's interesting that I just got my latest veterinary journal, and on the back was a big advertisement for yet another competing antibiotic that's supposed to get, you know, all of these bovine respiratory disease complex agents too. So the money is in cattle and swine. The money for testing is not even in domestic sheep. It's a minuscule industry compared to the cattle industry, and there's just no money to test it. So
But you
no,
do
there
say-
isn't anything.
You do notice results in 12 days or less, huh?
I'm sorry, what?
You notice results in 12 days or less?
You should be, they indicate that it is a one-time, because no producer wants to go back and have to re-inject, you know, his cattle once a day for 3 days or every 3 days. So if you can just hit them once, for severely affected animals that are not responding, it is labeled for use to be repeated in 12 days. But the intention of the antibiotic is to be curative, at least to the point of making the animal clinically normal enough to eat, gain weight, and move to slaughter, pasture, whatever, as a one-time treatment.
And you also have a tough time when you dart them, you can't identify them 12 days later.
Well, we have been looking at trying to take, we have some individual animals with horn markings, or some of the big rams that
Yeah.
people know, and we have been able to identify them, but that's one of the reasons why we'd like to now go in and get more collars and more IDs on these animals, because the animals that we have treated with Draxon so far are still alive. Doesn't mean that we cured them, but we might have made them feel better enough that they're able to get up on the mountain and get up and eat. But we want to make sure that we can identify as many of those animals and then the untreated animals as we can, so we can really get a good idea of, did it make a darn bit of difference in the end?
You see any big rams up there?
Don't you agree that, you know, with the, you know, what's in the cross results, you know, the ones that live, even with this pericardian inflammation and the lung sac adhesions, that even if they survive, it's going to take them a long time. I mean, if we have a bad winter now, we're going to lose a lot more, but it's going to take them a long time to lose those adhesions and everything,
Right.
everything else. So they're not going to be healthy to full speed for a long time. Do you
Right.
agree with that?
Yeah, and you know, I think that there probably will be some that always have some residual lung pathology left. And what's important to know is this a productive member of society? If it's a ram, can it become a trophy ram? Can it get the job done during breeding season? If it's a ewe, can she pull off a lamb and raise it to weaning, or is she a non-productive member of society? And so, because if we can't get these animals back up to healthy production levels and increase our lamb recruitment, then, then maybe Montana's right. Maybe we should just go in and eliminate everybody and start again.
And save money.
Right.
If that doesn't give you the information, if you're
I'll do the audience in a minute, turn the phone. Who else? Commissioner Morai?
Thank you, Mr. Chairman. First of all, I'd like to say that I'm impressed with, in the beginning, the way the public was informed of the event. The way the public has been kept informed throughout what was going on. In the face of disaster, it would be pretty easy to point fingers as to why this is going on or what caused it. That hasn't happened. And I appreciate the way that Bendow has handled this, and they deserve the credit for it.
Anyone else? I'll turn it over to the public. There was somebody had their hand up. You want to step to the microphone so we can hear you?
Yeah, Donald, with livestock, this time of year, you have moisture and things. If I was raising those sheep, I would be taking hay pellets out there, or not pellets, but the cubes, with about 50% grain, because in a feedlot, you have animals start getting sick and pneumonia. You have to raise the energy level. You do that with grain. And with these tub grinders, if you grind alfalfa and you go out and if it starts, you look at the animals out there, they start humping up their back and hair starts getting, looking bad. You start throwing, say, 200 pounds of corn in per every ton of hay, and it just snaps them right out of it. The sickness just flat goes away. And that might, some free choice minerals, and then another choice of some hay with just regular square cubes, and or they could be spread with a helicopter to be like browsing, like they weren't being fed, but the energy level is what is, I think, the problem.
Do sheep eat corn?
Hmm?
Will sheep eat corn and grain?
You bet.
Not so much on it.
Candy.
Thank you. Any other comments by the commission? If not, we have another comment from the audience.
Well, building Washoe County, I believe it all comes down to dollars, and we certainly need more of it, and biologists need to get in the air a lot more. I harvested a ram in Unit 181 this year. He's almost 7 years old. He lived through the die-off of 2 years ago in Unit 181. I brought pictures, I don't know if Mark shared them with you, of his lungs. He definitely lived through the die-off. Yeah, about 15 to 20% of his lungs were black with the adhesions. We also brought a ram out of lung tissue out of 183 where we had a small one. I don't know if you have the results back on that yet. Was he
He didn't have any mycoplasma.
no pastorillus?
Yeah, most of them have pastorella, but he did not
But
have
no
any
mycoplasma? And then one other question, 012, where we just took had the capture. Can we take anything out of their blood to find the selenium levels in that area?
We've, all that stuff is dribbling back in on the fax machine as we speak. And again, we were able to culture some of the common pastorellas out of those animals' lungs, but there was no mycoplasma detected. And just a cursory glance at their selenium levels, they're kind of mostly, again, inadequate to low normal to maybe slightly deficient, but mostly in adequate range.
Okay. One other thing, with the selenium blocks, placement is mostly by chopper. So it adds great cost to I mean, it's not just about going out and putting selenium blocks or what they're using is protein blocks. So the cost is very big when we go out and place these, or when the Department of Wildlife places them. We just need more money, guys. Thanks.
Thank you.
Well, while we have the biologist standing here, I saw Mike's in here and you, is overall, I had two questions. One is there any indication that you get resistance to the antibiotics with time, so that you've done something this year and you're gonna may have cured the problem this year, but we may not have something that works next year? Because most of the animals that get this basically go to feedlot or get killed. Do we know that this is gonna cause a problem with stuff? I think there was a question asked about mutations or something of the things, but I just wanted to ask that, if you knew that.
No indication, and I think certainly that, you know, with the feed companies and the producers and the drug companies are trying to get an antibiotic that with a one-time use does not produce any type of resistance and provides enough of a kill so that there's not residual organisms there to mutate and become resistant.
The next question I had included, Mike, just so I'm going to put you on the spot, both of you. We have the disease in Dawson, these Humboldts and the rubies up there. Overall, within the state, how much does this die-off impact the huntable herd for the future? I mean, as far as, I mean, Pat Cummins gave an opinion at our board meeting, and his answer was there was really little or no impact, even if we lost 50% of the herds up here for the huntable sheep in Nevada. And I just wondered if you guys had an opinion on that, what it would do from a biologic perspective.
Well, and you have more rams that die, fewer tags. We can talk a little bit about that tomorrow.
Right.
So we had over 220 tags statewide, and only a dozen or so Rocky tags. So, you know, if you just focus on the Rocky, not 30 tags, it's certainly percentage-wise, you might be looking at something that.
We can't, Mike, we can't hear you if we need to step up the microphone so we can pick it up. 'Cause I couldn't even hear the
Sorry, Mike, I
question.
need to put you on the spot.
Just, if we just look at the Rocky Mountain Bighorn tags, about a dozen.
We're talking about that tomorrow, right?
Right.
Yeah.
Paul was asking, will there be an impact? Well, it's all relative.
Yeah, well, you're going to report on that tomorrow, aren't
Right,
you?
right.
So we'll discuss that tomorrow. That's not.
Well, we don't have a crystal ball, what's going to happen over the next 3 months, and that's going to be the big crux of the discussion tomorrow.
I think that we can thank the department that we have a veterinarian on staff to do this kind of work and to work with the biologist. And we, in years past, we've never had this. We've had to farm it out to Davis and the results, and we weren't as vigorous pursuing these problems. So I want to thank you and the rest of the staff for doing what we're doing here. And with that, let's take a 5-minute break.